Great. I'm sorry, everybody. I know that was a very short break, but we're going to go ahead and get started with the last session for the day. And so we have another panel discussion lined up. This one will be moderated by Jeff Wenzel. So Jeff has worked for the Missouri Department of Health and Senior Services for 22 years, where he has served in a variety of roles, including laboratory scientist, environmental specialist, and epidemiologist. For the last 7 years, Jeff has served as the Bureau Chief of the Bureau of Environmental Epidemiology. Jeff and his team started wastewater monitoring in Missouri in May of 2020. And for the last 8 years, Jeff has been a member of the Interstate Technology and Regulatory Council's per and polyfluoroalkyl substances team. And in 2024 was chosen to be one of 3 co-leads for that team. Ooh, that sounds like a tough job. All right, Jeff, I'll turn it over to you. Thank you.
Thanks, Anna. Thanks everyone for coming this afternoon. We have 4 great presentations. When I talked to these panelists, they wanted to really talk to you guys and see what you were interested in and what questions you might have. So we have 4 pretty brief presentations talking about inhibition. First up, we have Errin Carter. Errin has a BS in biology with a concentration in marine sciences from Virginia Wesleyan. She is microbiologist at Hampton Roads Sanitation District in Virginia Beach, Virginia. Her areas of interest include public health and promoting sustainability through community outreach. The molecular lab that she works in has been using source tracking for a decade, and 4 years ago, when they began using that method and concept to implement wastewater surveillance, which allowed the molecular lab where she works to have the longest running uninterruptible COVID wastewater surveillance in the US. Her published work is redesigning SARS-CoV-2 clinical RT-qPCR assays for wastewater, RT-dt-PCR.
Thank you, and thank you guys for staying. I know we're the last session. So I work at HRSD, which is Hampton Roads Sanitation District, and I'm going to discuss things we've learned about PCR inhibition with more of a focus on the lab aspect. I'm a lab analyst. HRSD, it provides service to 20 cities and counties in southeastern Virginia and on the Eastern Shore. We release about 225 million gallons of treated wastewater per day, and we serve about 1.9 million people in our area. One thing I learned about inhibition is an ounce of prevention is worth a pound of cure. One thing I dislike in the lab is reruns. So if I can prevent them as much as possible, that's what I'm going to do. So I'm sure all of you have seen the overview of the molecular workflow. I'm going to give it a different spin, and I'm going to show you what I would do at each step to prevent inhibition. So we get our sample. I work at a wastewater treatment plant, so we're using the raw wastewater. And it might surprise you, sometimes it has different odors. It stinks regardless, but sometimes it'll smell like a chemical, or we have a lot of industries in our area. We have a lot of military installations. So if we see a sample and it has a strong odor or it's not going through the filter because we use an HA concentration step, we usually filter 50 mils of our raw wastewater. I would suggest instead of doing those 50 mils, just filter 25 mils into 2 different filters. Next, we do an extraction step. We do an automated extraction. Here we have a picture of our EasyMag extractor, and we have our filter, so we do an offboard lysing step. And if I put my filter into the lysis buffer and I notice it's a lot of particulates, I might go next door to our filter room and ask them, hey, can I just get a 2 mil aliquot? I don't even want a filter. Just give me 2 mil straight raw wastewater, because our extractor, it uses tips and cartridges, and those tips can get clogged, and that might fail on the extractor, causing me to do a rerun. Oh, one thing I didn't mention is if I had them do the 225 mil filters, we elute 100 microliters of eluate. So we're able to do all of our analyses that we want to perform. So I would have them give me the 225 mil filters and I would elute 250 microliters eluate and combine them at the end to bring the volume back up to 100 microliters. We also spike our samples with an internal control that mimics our analyte of interest. So if we're looking for a DNA sample, we would spike it with SCITA that looks like what we're trying to recover, or we'll spike it with an enveloped or non-enveloped RNA virus like hepatitis G or B. cov or bovine coronavirus. And we would spike our sample and then our negative extraction control, and we would use those numbers to get our percent recovery to see if it's inhibited. Next, if I'm on the pipetting step, I would try to see if my sample is discolored in any way or if it's viscous when I go to pipette it. That might give me an issue during my droplet generation since we do ddPCR. During that oil generation step, it can get clogged, and that's another step in the process that can cause inhibition. So I tried to prevent it during the sample filtration step. I tried to prevent it during the extraction step. I also tried to prevent it while I was plating, but by now it's on our reader. And prior to doing the reading of our samples, we have an optimization to make sure we have the best annealing temperatures for good separation when I'm trying to interpret the data. So after it's been on the thermocycler and the reader, we start to analyze or interpret our data. And if you look at the chart up there, you would see at the bottom, I have my negative baseline, and there's an area that I circled where it's slightly elevated on that baseline. That's something that would show me that there was possibly inhibition. So on the bottom, I circled it again, and I noticed when I was plating these samples that it was kind of thick when I was trying to suck it up with a pipette. So I circled that on the bottom that shows you how many droplets we have, and we aim to get about 20,000 droplets per sample. In the circled area, there's only 3,000 droplets, so it shows that my sample was inhibited, but I caught it and right next to it, I had space on my plate. So I just went ahead and I plated it at a higher dilution. So the first one I did at a 2x, and then right next to it, I did a 5x. And it's still low for droplets, but you can see it went from about an average of 3,000 droplets all the way up to 10,000 droplets. So the result could be calculated. And this is my contact information. That's it. Thank you.
Thank you, Erin. Next up, we have Thomas Clerkin. Tom is a research specialist in Dr. Noble's lab at UNC Chapel Hill, where he's been involved in WBE since the onset of COVID-19. He has played a pivotal role in collaborating with researchers, government agencies, and public health officials to develop a statewide surveillance program that currently monitors SARS-CoV-2, influenza, RSV, and emerging pathogens in 37 wastewater treatment plants across North Carolina. With expertise in molecular methods, digital and quantitative PCR, and assay design, Tom's recent work with NEWS focuses on training laboratories in optimizing wastewater processing methods, emphasizing high throughput capabilities and quality controls. Thanks, Tom.
Perfect. Thank you for that. Yeah, so I'm Tom Clerkin. I'm a research specialist in Rachel Nova's lab and a partner with NC DHHS and working with the NC State Lab of Public Health, and excited to share with you all kind of, as Errin just showed us, the value of using our controls and looking at inhibition controls for wastewater surveillance. So a little background for us. We all started in 2020 at the start of the COVID pandemic with the NC Pathogen Research Network, and it was a great collaborative group of academics, state and local government agencies, as well as some industry partners that we use to optimize these initial workflows and assay optimizations. What we did is we ended up with the HA filtration method that you've heard a lot about today. We acidify our samples and then add magnesium chloride to aid in viral adhesion to these NCE filters. While we were optimizing these workflows, we had a really strong emphasis on our controls. And so we had a control at every step of the process. So as we got our raw influent wastewater in, we spiked it with, as Errin just said, we did BCOV as well to match kind of the same viral characteristics as SARS-CoV-2, which we were doing at the time. And then we have a step for extraction control to see how efficient our extraction recovery is, RT efficiency, and then PCR inhibition efficiency. So then moving into 2021, we started working with NEWS and NCDHHS, and since then we've analyzed over 4,000 samples for SARS-CoV-2 variants, emerging pathogens of interest. And then currently we're working along with Bio-Rad to optimize some 6 target respiratory panels on the QX600 and looking into different influenza panels for subtyping, AMR targets, and then GI panels. So why we're here to talk today, PCR inhibition efficiency and our controls. What we're looking at right now is monthly average controls at 2 very different wastewater treatment plants in North Carolina. So Beaufort is a treatment plant with less than 1 million gallons of discharge per day, where Raleigh is closer to 50 million gallons of discharge today. With that top graph, that's going to be our PCR inhibition control. And as you can see, it's really consistent across months at these 2 sites that are very different, have different inputs, different organic materials, different inflow and infiltration rates. Yet we see really good PCR inhibition efficiency. We run this control on every single sample we have, and we average about 95%. But having this like really big longitudinal data set of how this PCR inhibition control works lets us know if we're 2 standard deviations away from kind of that average that we're seeing at, we need to go back and do some inhibitor removal steps, some dilutions or something to remove the inhibition so that we can be really confident in our concentration data that we're generating and to avoid false positives for these new emerging low target pathogens that we're looking at. And then this bottom graph is going to be that total processing control that I was just talking about. And with this, you do see much more variability in this kind of, yearly transect look at this data. And it does kind of seem like there's a little bit of seasonality to it, which, we can speculate is from different rainfall and organic materials and entering these different wastewater treatment plants. But then again, we have this really good longitudinal data set where we can go back and look to make sure that the data we're generating and the concentration results we have are good and providing us impactful public health data. Again, going back to that longitudinal data set, by having this, we're able to optimize new methods as we look for new pathogens. A couple people have talked about today how we, potentially need to optimize new methods for certain targets. And so when we were starting our flu and RSV panel, we saw some papers that saw 1,000 times increase in concentrations in the solid fraction versus the liquid fraction. And so we worked quickly in the lab to optimize the solids methods. And the first thing we did while optimizing that method was to look at our controls. And we saw our controls were looking similar to what the kind of gold standard was for the state of North Carolina, was that liquid influent HA filtration method. So since we had that, similar, comparable, or even better, we were confident in the method and we can go forward with those samples. And then finally, in North Carolina, we're about 50% rural. So we have a lot of rural wastewater treatment plants. To increase our coverage in the state, we're going to have to pay attention to these rural systems. And so we have kind of some pilot studies working on this in Eastern North Carolina at some package treatment plants locations. And the pathogen signals in these locations are really highly variable. And it is kind of because there are less people, more variable inputs coming in through there. And so as we're looking at these rural wastewater treatment plants in North Carolina, we need to pay really close attention to our controls to avoid false negative results and what we're looking for. And with that, thanks, everybody.
Thank you, Tom. Next, we have Dr. Chung-Ho Lin. Dr. Lin is the lead scientist for bioremediation, natural products, and bioanalytical programs at the School of Natural Resources at the University of Missouri. Since the COVID-19 pandemic in 2020, Dr. Lin has joined a task force sponsored by state and federal agencies to monitor the prevalence of human pathogens and illicit drugs using a wastewater-based epidemiology approach. He has successfully utilized modern I looked at this word a couple times before coming over here, metabolomic techniques to identify bioactive chemicals that can quickly suppress qPCR signals in the wastewater.
Yeah, thank you for having me here. It's such an honor to get a chance to meet a lot of scientists and also government agency. We are sharing the common interest of wastewater. So based on my experience, there are two major sources of a signal suppression. Okay. First is a chemical in the wastewater that immediately degrades the lipid bilayer of viral particles. The retention time is about 24 hours. Assume majority of us are using this 24 hours composite sample. That means those chemicals will have 24 hours to break down the viral particle that we're interested. And this is an irreversible process. That means once they degrade, nothing we can do about it. The signal cannot be recovered. Another source of a signal suppression is during that PCR reaction. Those chemicals in the extract that interfere, for example, the polymerase, or it might bind into the dye, or it might bind to the template. Usually that reaction time is about 2 to 3 hours during that PCR reaction. Fortunately, there are several strategies that allow us to recover the signal, such as a simple dilution with buffer, or there's several dilution technique in the instrument design, such as partitioning, right? For droplet digital PCR, they partition your extract into 30,000 of small droplets. In that process, that dilute the PCR inhibitor, or such as microfluidic digital PCR, right? You dilute your extract into 25,000 physical small reaction well. That will help to dilute the inhibitor. So in this case, the signal can be recovered. I know that some other group also use the column, right, to remove the PCR inhibitor. So in that case, a lot of signal can be recovered using different strategy. I have invested almost 3 years of my life focus on how to go back.
Yeah.
Focus on the first one. So I did what
That one?
okay. Focus on the identify what class of chemical in the wastewater that can degrade the viral particles. What will be the implication? What will be the mitigation effort? Hopefully, now we're hoping by quantifying different class of a chemical suppressor, we're able to develop the criteria to design the algorithm and the correction factor to allow us to calculate and determine the copy number in the wastewater in more accurate fashion. So how can we identify what class of compounds in the wastewater that degrade the signal immediately? How many of you remember in the high school using the filter paper, right? To separate different classes of pigment. Same principle, okay? We're using liquid chromatography packed with resin. Then we load the wastewater. We know has shown the strong signals of suppression. We load them on the column by manipulating the solvent gradient. Every compound will travel at a different speed based on that hydrophobicity. Basically, we separate by the hydrophobicity. Then we set up the fraction collector. Then we collect every fraction. Imagine you start with 10,000 chemical. Then after fractionation, you separate different kinds of compound based on their chemical physical property. In this case, we start with one fraction we know has shown the strong signal suppression. We end up 31 fraction. 4 of the fraction we test with the wastewater with a known copy of N1, N2. We know, right? We start with probably 10,000 chemical. We end up only 20 compound into each of these half fraction. So what will be the next move? We know those are the half fraction with a compound of interest. Then we inject this half fraction into the high resolution mass spectrometer. With the current analytical capacity, the high resolution mass spectrometer, just as one single injection, they will allow us to identify quarter millions of molecules. So every compound, there is a fingerprint. We call fingerprint. We call the mass spectra. We compare this fingerprint with a reference fingerprint in the library that allow us to quickly identify those compounds. Especially a lot of these compounds, they have been very well known, characterized as a biosurfactant emulsifier. So then we integrate this metabolomic platform. They will do the signal detection, peak alignment, peak annotation by comparing with this we call the mass spectral library, the Malin library, one of the most comprehensive mass spectral library on Earth at this point. So we can quickly identify those bioactive compounds in every half fraction. Then we purchase a pure reference standard for target analysis, for validation, also allow us to design the degradation study to understand the degradation kinetics. So far, we have identified 54 surfactant emulsifier and also several active ingredients in the disinfection products. Then we're hoping using the algorithm to allow us to design the criteria. Then we can either develop the correlation factor or we can exclude some of the facility from the at the very beginning of a surveillance project. So this concludes my presentation.
Last but not least, we have Reinke. Ryan's from Los Angeles County Sanitation Districts. He spent the last 21 years focusing on investigating key topics in environmental bacteriology related to wastewater treatment and public health. His work at the Los Angeles County Sanitation District has included research activities such as wastewater surveillance, studying the effects of wastewater treatment on antimicrobial resistance, detection of enteric viruses in water, assessment of rapid indicator methods, biological nitrogen removal, determining the fate and transport of viral and bacterial pathogens in wastewater, and studying wastewater disinfection procedures.
Thanks, Jeff. I'll be quick. I think some of the panelists have covered most of what we've talked about inhibition. But I will present a little bit of our wastewater surveillance work that we've done at the LA County Sanitation Districts with a focus on inhibition. So a little bit about the organization, and I'll be quick here. We are one of the larger wastewater organizations in California. We operate 11 wastewater treatment plants. 10 of those produce reclaimed water that's beneficially reused across the region. Each of our plants are kind of speckled with or identified with the blue dots there across the LA region. We collect and treat wastewater for roughly half the population of LA County. So our SARS-CoV-2 surveillance began in April of 2020 and continued through May of 2021, at which point we then transferred that over to state and commercial labs. So the data I'll be talking about today is really the beginning of the pandemic. We analyzed influent from 2 of our larger wastewater treatment plants. They service about 4 million residents across the greater LA County region. We employ a direct extraction method, as we heard from some of the previous speakers. That involves basically directly extracting the RNA from a wastewater sample, and then we analyze by RT-qPCR, although we've now gotten into the digital era. We tested several different RNA extraction kits. The Zymo fecal soil microprep kit won out, performed best with our matrices in our hands. It doesn't include an inhibitor removal column. Now, the way this is a depiction of our workflow, and really I just wanted to highlight a couple things here. The way we dealt with inhibition was kind of a multi-pronged approach where initially we added in we used an in vitro transcribed RNA that was added into the samples and the mock extractions in equal amounts, processed and quantified by RT-qPCR. And then later on, as the OC43 virus became more readily used in the field, we incorporated that as an additional sample processing control, which gets added in at the point of sample collection, follow through, and then again quantified by RT-qPCR. And then finally, as a final kind of assessment of inhibition, all the samples get run with 3 different dilutions: undilute, 1 to 2, 1 to 4. So again, just kind of a multi-pronged approach to try to identify inhibition and issues with the sample processing. Really briefly, this is just a summary of some of the sample processing and inhibition control data that we saw over the year or so that we were monitoring. Generally, we got recoveries above 70%. That was for both the in vitro transcribed RNA or the OC43. When we looked at assessing inhibition, we're looking at taking the difference in cycle threshold values from the samples and the mock extractions. It was pretty consistent. We didn't see a lot of inhibition there. Over the sampling campaign, the average difference was less than half a cycle for the in vitro transcribed RNA and slightly more for the OC43 at about 0.6 cycles. But in general, what that's saying is there wasn't a lot of inhibited samples. When we did occasionally see some sporadic inhibition, both controls picked it up at about the same percent. About 5% of the total samples were inhibited. The 1 to 2 and 1 to 4 dilution series that we ran tended to abrogate most of the inhibition that we saw. And that dilution series was also helpful in identifying some additionally inhibited samples that the 2 process controls failed to detect. And so, and there's nothing groundbreaking here, but factors that we saw that affected the presence or absence of inhibition were the choice of matrix. We did look at some primary solids initially, had higher levels of inhibitor than the influent. The extraction methodology mattered. Generally, extraction kits that included an inhibitor removal step performed better than those that didn't. And then finally, that dilution series can be helpful not only abrogating inhibition, but also identifying some additionally inhibited samples. So with that, I'm going to conclude and we'll get on to the panel discussion. Thank you.
All right. Thank you all. Happy to take any questions from online or in the room.
All right, we have one online question here, and it's for any of the speakers, really. How did labs settle on using bovine coronavirus as their viral controls as opposed to using heat inactivated SARS-CoV-2? And would labs consider switching to HISCV2?
I guess I can go ahead. So we have done both. We've done the BCOV and spiking in some SARS-CoV-2 in our samples. And to avoid potentially increasing the concentrations that we're getting, we wanted to use a different target than what we were analyzing for these different WBE studies.
With our lab, we test a lot of different analytes, and being able to use BCOVE, it lets us, I guess, get the recoveries of not only coronavirus, we can compare it to like BRV and different viruses. So it's more cost effective to use BCOVE in our situation.
So how, as we're looking at the data, how could we tell that we might have some inhibition issues either in the lab or Dr. Chung-Ho from our samples?
Yeah, we have been using the one engineer internal standard, the pure with that unique sequence that cannot be found in the nature to evaluate whether that we have a PCR inhibition or we have that source suppression. That's one way to help us distinguish.
How about for the rest of the panel for within the lab where we would have inhibition issues?
Yeah, I think it's you know, kind of the what most people already know is that as often you know, at each step, if you can provide a control, I think it provides a little bit more confidence to the overall data set, and that you're the numbers that you're generating, you have a little bit more confidence in the fact that it's not being inhibited or you're not losing it at some step during the sample. So you know, I think as Thomas said, you know, as often as you can control at each step along the way, I think it just provides more confidence in the data overall.
Do you know how much difference the inhibition can make? If you've maybe some of the examples that you've seen, worst case, how much difference in the signal?
Yeah, we've seen complete total inhibition all the way to next to none. You know, treatment plants are, as I think most people know, that each one is a beast in and of itself, and even, you know, throughout the day, what comes through the plant can change pretty dramatically. And so from sample to sample, I think at least the approach we tried to take was to try and identify extraction kits and workflows that worked in general, but every once in a while something comes through the plant that you just most of the time don't know. I mean, maybe Chung-Ho can help us identify some of the inhibitors that are coming through at different spots. But yeah, we've seen complete inhibition where we even diluted out, you know, 100-fold or so. It still was inhibited.
Yeah, so in North Carolina, we had a couple instances at 2 plants in the state where this has happened, where we were doing twice weekly sampling. And so one of the samples would be great. We get a high concentration of SARS-CoV-2, our controls look great. Then the other sample collected on the same day, every single week, would always show up with like 1% recovery and 50% PCR inefficiency. And I think Brenna said it perfectly earlier, the lab's going to riot if you keep changing all your methods. So what we ended up doing was we talked to the treatment plant operators, and they were identify some industrial inputs that were coming into the wastewater treatment plants. One of them was some discharge from a drinking water recycling facility, and then other was a chemical input. And so kind of having those controls give you that kind of adaptive capability to go back to your treatment plant and ask them to change their sampling methods or days in our case.
This one's for Dr. Chung-Ho Lin. How would you go about developing a correction factor to account for inhibition?
Yeah, with the access to 55 facility in the state of Missouri, where we saw this suppression effect, we've been comparing all the chemical profile across a different facility. We identified there are the 54 the surfactant that have been commonly found in the wastewater that provide us a strong indication. So we've been trying to developing this algorithm, hopefully, by quantifying these 55 surfactant in the wastewater, we can immediately develop this correction factor, to evaluate how much signal was suppressed during that 24 hours sampling time. Yeah, right now we already have an analytical method, basically just one injection. All we need is 1 microliter. We can quantify all these 54 the bioactive compound within 15 minutes.
So just to follow up, I find that metabolomics approach quite fascinating. And we've also been trying to understand how we could use mass spec to look at inhibition and of course, some of these targets. But I'm curious about narrowing down this group of 54 or so surfactants. Is this a group that you consistently see across different treatment plants, different maybe manhole samples, sewage samples collected from manholes?
Yeah, it depends on nature of the industry within each sewer shed. We found that particularly for sewer shed where they have this food processing, facility, usually we found an extremely high level of this surfactant in the wastewater. Then usually that degradation rate, I would say more than like 60%. We did like a time course kinetic. Within, I think 60% of signal will be immediately suppressed over the first 3 to 6 hours. But of course, there's synergistic effect. So by knowing their environmentally relevant concentration in wastewater, we published a paper on that. So we create a cocktail, try to simulate what is the synergistic effect that provide a first step, a foundation, a solid evidence, what kind of suppression rate during that first 24 hours period.
And just going off on a slight tangent, can you detect the N protein by mass spec?
Protein, yes. Actually, this mass spec is designed for small molecule, but in order to quantify this long chain peptide or protein, we can use a digestion. If we can map this amino acid sequence, identify that unique sequence, we can, through this digestion, we can make that small piece, so that will fall within the range of detection. We can quantify that. At one point, we are thinking about using the mass spec to quantify the neutralizing antibody in the wastewater. That will give us another layer of information to assess immunity of a community. That's still work in progress.
Yes.
So my question has to do with a factor that some of us use for normalizing our data, and that is a fecal measurement like PMMOV or HF183. Those are in very, very high concentration, maybe too high for using the same dilution or the same concentration of the sample as we use for SARS-CoV-2. So those samples are diluted 1 to 100. Dr. Reinke noted that sometimes the inhibition isn't even apparent until you do a dilution, so you might not know about it. How do you handle, diluting 1 to 100 or 1 to 1,000 for normalization if you're not sure whether there's some inhibition in your supposedly uninhibited sample that you just laterally discover has some inhibition?
Can you say the question again? I was when you
sorry.
say uninhibited sample, what are you talking about?
Well, the sample that you get out of your purified material, you go ahead and do an assay and you get a certain number. If you're making a fecal measurement, you might take that concentrated sample, dilute it 100 or 1,000-fold, and use primers for that. But that would have any inhibitors that was present in your original concentrated sample diluted out, so there'd be less inhibition. So you're not necessarily on every sample normalizing in the same way. In other words, should you multiply by 100 or by 1,000, or is there something that you do to evaluate what the change in inhibition is?
Yeah, I see what you're saying. So we, with our sample dilution, it is, the final extracted RNA is what's getting diluted prior to going into the reverse transcription reaction. The control sequences are usually there in high enough amounts that when we dilute down, they're still detectable. So it's not an issue of I guess whether the primers would be getting diluted out or not, but anything that comes through the extraction process is what we're diluting out. We generally don't like to. I mean, I guess there's two sides to this, but we generally don't like to add correction factors if we don't have to, because it gets into some problems about how valid are those correction factors and what do you correct for? Is crossphage better than PMMV or something else? And so generally, if we have diluted out to 1 to 100, we feel pretty confident in going, okay, well, if we find something there, it's going to be at least 100-fold, higher concentration, or we'll multiply directly with the dilution factor.
And yet CDC only accepts data, as I understand it, if it's accompanied by flow data or a fecal measurement, even though many of us don't use that fecal. We measure it because we have to. The state says that way we can submit it, but we don't use that number to normalize either.
Yeah, we would agree with that.
All right, so this one is for any of the panelists. Can recovery be used as a quick and dirty indicator of inhibition? It wouldn't require any additions to the workflow.
I could start. We kind of started out doing the same, using kind of a process control as recovery control and inhibition. And for us, and we have very limited resources when we were doing this, so we kind of had to expedite and make it adapt our program to what we had available. So we basically said if it was below a certain level, if it was below 70% recovery, regardless of that, whether that was due to the recovery or the inhibition, we reran the sample or just, the data we deemed as not reliable. And we didn't go beyond trying to figure out, well, was it a recovery issue or was it an inhibition issue? We just said, okay, it was too low based on one of those two scenarios.
Yeah, I think that's a really good question. And going forward, as we're looking at new emerging pathogens, with our 6-color panels that we plan to be doing, 2 of those channels are going to be taken up with controls. So we'll have our PCR inhibition and our BCOV on every sample. And then kind of going back to that last question about having to do the dilutions with PMOV, that's kind of how I see a benefit to this total processing control, because you're going to spike that into your sample at the concentration that you kind of expect for these targets of interest that you're looking for. So I think you bring up a really good point about the PMOV as kind of a difficult correction factor to use.
Any more questions from anyone? Thank you, panel.